Well, good afternoon. Thank you for being here. This is the last session today. It's very important for us being Bactools. We are a young company producing bioinputs. I'm really honored to share this panel with Gustavo, whom I have known for many years. So thank you for inviting us and giving us the time to address you. I will tell you what our company does. We are a biotechnology company producing bioinputs, and we have a strategic partnership with the partner Aapresid. Aapresid is a company producing traditional fertilizers, nitrogen-based fertilizer. We have at Bactools, we have a clear idea and a clear focus on plant nutrition. We also work on animal nutrition. Our vision is to improve has been used as a tool in agriculture for some time. Now we believe there is a long way to go, and we want to maximize the synergies between bioinputs and the current agrochemicals to improve nutritional efficiency. Basically, our vision is to work with the microbiome. We believe the microbiome as a consortium is very important, and it has a high impact on the soil and the crops. And there is also a synergy with agrochemicals. How do we work? We use a technology called the technology of spora of the Bacillus family. These spores have a specific feature. We prepare reagents and we produce spora that are stronger than the bacteria we already know. So these bacilli, as they are bacteria, at a given point in time create a spora which is quite resistant. We are able to develop products which have other features. Without talking too much about this, we work with the bacillus bacteria. They create spora, and at a given time they grow in a vegetative fashion. They grow, they split, they grow, and at some point in time these cells create an endospore. This is a microscopic view. The black bananas are vegetative cells and the other ones are spores. This is a picture of an taken with an electronic microscope, and you can see the characteristics of a spore. You can see a coat, a cortex, a cell wall. The DNA is quite packaged, it's dehydrated, and this structure creates a resistant spore. It's resistant to heat, radiation, drying, and corrosion. Many environmental factors. Therefore, we are able to develop more resistant products. We work using bioreactor, and within the bioreactor, in controlled conditions, we create spores that are quite resistant so that they may be combined with the products I will describe soon. Going back to our vision that is improving the tools we already have in the microbiology area today and maximizing synergies with existing agrochemicals, we developed several products. So bioinputs, are they better or worse than the traditional agrochemicals? At some point in time there was a discussion whether one group or the other was better. People have said, I have used bioinputs and the results were not good. I've used them and I can see no treatments. They are not as good as the traditional chemicals. So we go against those paradigms because we believe those two inputs have to work together. It's not either or. Both of them provide benefits. You know about the benefits of the biochemicals, but we understand that bioinputs complement and add something to the traditional inputs. And they provide many benefits, especially in the area of nutrition. These are the experimental results achieved with our consortium to talk about the contributions of bioinputs. We have developed a consortium which we have named B4. It's made up by 4 bacteria of the Bacillus family, and you can see the names on the slide. And together, this consortium was used with other technologies such as bradyrhizobium. We have used brady with these 4 bacilli, and the same consortium of 4 bacilli was also used with another product called AgriBio. AgriBio is a fertilizer used in furrows. It was developed by Agrifer. We are using liquid fertilizers with 10% of the bacterial consortium. We reduced the use of nitrogen fertilizer by 10%. So finally, we will talk about the effect of a bacteria. A simple one, it's not a consortium or a family. It's a strain of Bacillus amyloliquefaciens, and we will see the yields. We will have a look at the yields. The first trial with Consorcio B4 was done in the 23-24 season in soybean, 12 repetitions, 4 sites, 48 treatments, and the results are there. We did the trials in Payamonte, Villa Maria, Rojas, and Balcarce. In this trial, we had a co-inoculation with bradyrhizobium, which is a well-known technology. Generally speaking, the yield was higher than that achieved with a Czech standard Czech plant, which was bradyrhizobium on its own. These are the 4 sites. Generally speaking, we always had good results, above 5% using the old technology, adding our Consorcio B4. This was a return on investments. The investment rate per hectare is in red, and in blue we can see the ROI when the Consorcio B4 was added. This happens when you stack technologies. Well, so we did inoculate just with brady, but with brady plus Consorcio B4. This is the proof of concept. This is a trial, 12 repetitions in Rojas. Again, we did a statistical analysis, and we worked on this with Agrifert using their product, the Surco fertilizer, and we designed Surco plus Consorcio B4 in what we called AgriBio, or AgriBio. This is a trial with wheat. This is a real picture made with a drone. These are small lots, 20 square meters, and you can see the results of the treatment. And what we were able to do in this case was increase yield by 8% with the fertilizer plus the microbial consortium. The fertilizer plus the consortium that improves the bioavailability of the fertilizer. And finally, the BSP trial, 4 repetitions, 3 sites. These are sites with different agroecological features, Piemonte, Villamelia, and Rojas. We assessed a strain that solubilizes phosphate in the trial, which is difficult to explain, but we will give it a try. We looked at the inoculation of seed inoculation, and what we assessed the yield without fertilization, with fertilization, and what we realized, considering the proof of concept we had already done, we looked at the possibility of decreasing the use of phosphate fertilization by 20%. We decreased it by 20%. So without fertilization, with the bacteria, with 80% of the suggested fertilization rate, with the bacteria with 80%, and 100% of the fertilization suggested by the consultant, plus with or without the bacteria. Here you can see the yield as the trial progress. This is the check stand in orange, trial number 1. And at the top we have the yield, average yield of the treatment. This is inoculant with the phosphate solubilizing bacteria this is without inoculant, this is with inoculant, this is with 80% fertilization and the inoculant, and this is, the speaker corrects himself, 80% is no inoculation, this is 80% of inoculation with inoculation, this is 100% of fertilization, this is 100% of fertilization with inoculant. That is, treatments 2, 4, and 6 include inoculation. Treatments 1, 3, and 5 do not include inoculation. So as fertilization increases and the bacteria is used as inoculant, we can see there is a better bioavailability of phosphorus in this case. Now, if we compare 4 to 5, we can see 4 is 80% of fertilization, of the suggested fertilization rate with simple superphosphate plus inoculant. On the other hand, you have 100% of the suggested fertilization without bacteria. And it's a 20% difference in fertilization with a very similar yield. So we might cut down on fertilization with phosphate by 20%. At the bottom, you can see the ROI. In red, we invest in this new technology, and in blue, you can see the increase in yield. How does the improvement take place? Well, this was published in Plant Science, but what is said is that inoculation with bacteria stimulates the roots. And we have found this experimentally, and you can see this in our booth. We have this place in our booth, and we have bacteria that cause roots to produce more lateral small roots, and we can see how the bacteria grow as the roots grow by creating a biofilm. And in this interaction between bacteria and the roots, many things happen. This is difficult to understand, but well, if we work with fertilization, this is the root, and fertilization of the Surco trial, you have the nitrate, ammonia, this goes to the root through the high and low receptors, as well as the ammonia transporter. This goes up, and after this we have biosynthesis, photosynthesis, and the same molecules, if we look at this side, the same molecules that came through fertilization move through the bacteria first do ammonia fixation, there is a synthesis of amino acids, glutamine, asparagine, and glutamic. There are specific transporters for these amino acids in the roots. They come from these interactions. There is an interaction between ammonia and the nitrate of the synthesized bacteria. We have cytokines, and different elements that stimulate the immune response of the plants. They are receptors for surfactants, and without talking about this in detail. Well, look at this. All the nitrogen, which is all this nitrogen, goes into the plant. So a bacteria working with a plant can do many things. It's very positive.
Los resultados, en líneas generales, los biológicos a nosotros nos están dando entre 6 y 8% de aumento de productividad a través de la mejora de nutrición, que si bien nosotros no lo vimos en este caso, pero a través de la solubilización de nutrientes, fijación de nitrógeno, la producción de fitohormonas, la producción de metabolitos secundarios como sideróforo, antibióticos, enzimas, eso es muy importante. El tema del biocontrol de los bacilos es muy, pero muy importante. La inducción de resistencia sistémica, vimos el tema de los sideróforos, todo este tipo de cosas son muy importantes y surgen a través de la interacción de bacterias con las raíces de las plantas, ahí en el microbioma. Y esto tiene altísimo impacto en el estrés abiótico. Imaginen una situación de una planta en estrés abiótico. El estrés abiótico cualquiera, o sea estrés hídrico, calor, oxidativo, cualquiera de ellos. Si la planta, además de estar en una situación de estrés, tiene que ponerse a sintetizar todos esos compuestos que nosotros vimos antes, o sea, es una pérdida energética enorme. Entonces la planta necesita guardar la condición energética, la poca condición energética que tiene en una situación de estrés para funciones vitales, y en ese caso, si la planta tiene un buen microbioma, ese microbioma va a poder nutrir a la planta sin que la planta tenga que gastar tanta energía en eso. Bien, para finalizar, nosotros tenemos la visión de la complementariedad de los biológicos con los químicos. Eso para nosotros es fundamental. Ni uno ni el otro. O sea, yo entiendo que por ahí puede costar adoptar este tipo de tecnologías, pero creemos fuertemente que la incorporación del biológico va a ayudar un montón a la incorporación de los químicos. Sobre todo en la nutrición. Estamos hablando, haciendo foco en nutrición. El uso de bioinsumos trabaja lógicamente en reducción del uso de químicos sintéticos, mejora la salud del suelo, protección de la biodiversidad, reducción de la contaminación. Estas son cosas muy, pero muy importantes, sin entrar en, por ahí, huella de carbono y un montón de temas que también son muy importantes. Y que es importante tal vez tenerlo en cuenta, porque imaginen que si uno puede reducir un poco la cantidad de nitrógeno que se utiliza.
If we reduce the amount of nitrogen into the soil, a lot of nitrogen is lost. 50–60% it is lixiviated, evaporated, or if we can reduce the amount of nitrogen as fertilizers, because the fertilizers that we use come from the Haber-Bosch process. They come from India, which takes approximately 10% of world energy to fix nitrogen from the atmosphere in ammonia. A nitrogenase compound takes approximately 16 RPP to fix one single molecule. It's very, very expensive. Nitrogen fixation is very expensive. The Haber-Bosch process, and I branch out completely. The energy that is approximately 300 degrees and a lot of pressure to fix the atmospheric nitrogen is the heat of fossil fuels. The emission of CO2 is very high. So having biologicals in combination with chemicals and the nitrogen input is very important. And I'm not talking about the Green Pact, the 2030 Agenda, because these two documents contemplate these things. What are the challenges? Argentina is one of the leading food producers all over the world. Reconverting this technological matrix by adopting this strong use of bioinputs is very important. And it's very important to become a country adopting this technology and developing the technology. The key bioinput companies are local companies. Baxter, Terragene, to name a few, Spunaview. We are talking about local companies, and this helps to the development of the local economy. And why is it very important, the fact of biomass. This is an engine that looks for scientific literature, and it's very important. Of course, we can't read anything here, right? But as from 2019, research and reports increased about the importance of biome. And I can spend the whole afternoon talking about this. And this is a snapshot of bio inputs, adoptions. This is a report by McKinsey 2023. In blue, currently using, light blue, planning to use, white, not using, not planning to use, and light blue, never heard. Brazil, for example, more than 50% has adopted this technology, biocontrol, biofertilizers, etc. So Brazil, almost 50% of the farms have adopted this technology, while Argentina, 4%, 11%, 6% of actual use of bio mass as a technology. Is there a resistance to using? Well, we have to overcome that barrier. Thank you very much. I had a lot of information to share. Thank you very much. And I give the floor to my next panel member. We are sticking to time. Gustavo González Santa. I was born after Haber-Bosch, because that was in 1945, and I was born in 1962. Just for clarification, right? Because it's I'm 30 years older than Walter, so just to give you an idea of the time frame, right? Overcoming barriers. So we have a problem. We need to overcome a barrier. So the outlook about adoption of new technologies on the part of farmers. When Nico proposed this topic to me, I started thinking about how to approach this topic. And basically, as we do as part of the academia, I asked myself 3 questions. The first question is, what is the technological scientific evidence about bio inputs? There is a lot of information. Science provides a lot of data, and this has been proven. There are many papers that have been published. So it was important to have the scientific foundation. I'm going to share some information. So there is a barrier for adoption, but there is something positive. The scientific evidence and the technological evidence is telling us that these type of products contribute and add value to our production systems. The second question I asked myself was, why do we have these barriers if we have good information? Why is it difficult to adopt this technology if the results or the findings indicate that we have positive yields and positive development of biomass and other things? And to put this, or to frame this, I asked myself about the conceptual framework, the current times we are living, because it's okay from an agricultural farmer point of view. We have a huge global demand for products or food. That is to say, our customers at the end of the day are ordinary people that need to eat healthier food. So based on that, based on Nicolas' unethical proposal, I tried to answer these three questions. So going to the framework, we all know that people want to eat better, healthier food. And it's okay also the climate change. We know that the climate change is here, but now we need to consider the impact, whether the temperature is going to increase by half a point or 1 degree or 5 degrees. And the third thing about biological inputs is stress management, abiotic stress in particular, because we have chemical tools for biotic stress, fungicides, etc. But if we go backwards and check climate conditions and rainfall levels, both in the country and abroad, abiotic stress impact has been very significant over the past years. The 22-23 season in the humid pampas, there was no soybean. People were not able to sow soybeans. So the first aspect, in addition to data, people are willing to pay for this. So as farmers, we need to deliver and provide or meet consumers' needs. This is a survey conducted last year, 2023, in Europe. We are talking about a market of 500 million inhabitants. Europeans are willing to pay more for better quality, traceable products that show that they have no negative agricultural impact. The second aspect, and this is bad news for you because there are many congresses going on, Paris, Beautiful City, 2015, Climate Change Summit, but if we analyze January's data, the CO2 curve is still up, methane, nitrous oxide, but it's going up anyway, so climate change is here to stay. We have to accept it. And the third aspect is that we are faced with new problems, particularly. I took this picture, so I apologize if they are not good quality, but this is a picture of a Paraguayan Chaco that I took in February. Go there in February, approximately 45 degrees Celsius. It is like a punishment to be there. And these are peanuts. And the difference between these three plants is that the first one didn't have a microbiotic consortium, and the other two, yes, they promoted root development, making water capture more efficient and taking advantage of rainfall water. So the bugs can help us a lot with regard to abiotic stress management and turn it into biomass. And on the other hand, these are three fungi. We need to change our mindset to apply an insecticide because we see some aircraft spraying the insecticide at 3 p.m. I know that you don't do that. That happens only in my farm. But this is Bovaria, Metarrhizium, and Isaria that are controlling bugs. And how the herbicide was applied, well, we did a foliar application, and the restriction is that we need to apply it with relative humidity higher than 70%, but we can do it. And these two issues, shortage of water, water management, and shortage of resources, and the leaf hopper, these type of strategies can help us to mitigate these problems. So the overall framework help us to promote these type of technologies because they add value. There is no problem with the demand for final products. We have inputs and biological tools that allow us to overcome these problems. Now, the question about adoption. If this is so good, why is there reluctance to adopt them? And among other things, it seems that we are undergoing disruptive times. 30 years ago, the first Aapresid Congresses, we challenged the no-till seeding or the growth of wheat or the fact that we have to till to have a better bioavailability of nutrients. But we adapted to the changes. But there is a concern about the use and management of live microorganisms. Because if I use a specific agrochemical, I know that I will have a positive result. But when we talk about live microorganisms, it is an issue. Compatibility with chemicals is another issue. And the adoption of this new paradigm, it is complicated. But in the 20th century, we are talking about 40 years, 24 years had elapsed, because Walter was talking about these bugs or the leaf hopper were discovered in the 19th century. So they are almost friends. But what changed during this century? And we can get better results of biomass and control of diseases and of insects. What happened? The formulations changed in these 24 years. So today, the relative weight of technology is greater than science, not because it is higher, but there was a physiology, morphology, and interaction curve of the microorganism and the plant interaction. We know that we can mix it with a fertilizer in the seed and we can apply it on a foliar surface and have a positive result because we had the appropriate formulation that keep these microorganisms alive. And when we use a biofungicide, for example, we are using microorganisms, and at the same time the metabolites produced during the production process of the microorganism. We were talking about surfactant, fenhexine. These are weird molecules, but in the 90s or earlier, we were talking about triazoles. Surfactants is like triazoles back then, but instead of using a chemical reactor in the molecule, I use a cell that produces a substance that has the same control effect to control a disease or to control an insect. So what has changed basically is the formulation, and the level of adoption has increased, although we need a faster adoption rate. The good news is that when we see the growth rates, let's talk about or think in the short term. Between 2018 and 2024, with biostimulation, biofungicides, and biofertilizers, there is an increase, and the trend is to continue going up over time. Why? Because there is an increasing trend to reduce availability of active ingredients. This started in Europe, then in the United States, and now in Latin America. So you can see that
in 10 years, we cut down on the use, on the amount of active ingredients. So the biological or alternative solution has to be used. And when we go to other area, and we look at the data in Latin America and we think about what is happening in Brazil and Argentina, we can see there is a growing trends towards the use of these alternatives. Maybe Brazil is taking the lead here because of their agricultural system. They go to the plots many more times than we do. Who goes into their plot 5 times to provide the soybean treatment? 5 times, none of us. In Brazil, 5 times at least, because they have soybean rust, lots of pests, many diseases. So this gives them a strategic opportunity to use biologicals when they are really necessary, when the disease appears. But we can see a growing trend anyway. And in wrapping up, I ask you a question. Would anybody be willing, considering the price of urea today is to use 560 kilograms of urea to produce 3.5 tons of soybean? Or either you are all asleep, this is one possibility, or no. Well, this is what we are getting. And it's interesting to mention this paper written by Alejandro, who is here in the room. He became a grandfather a short time ago, but he did this with other researchers, and this is clear. Look, see for how many years we have been using a very interesting resource that helps us to increase yield by 8%. So this bio-input, well, it's crystal clear that we have to use it, and the efficiency is quite high and it stays a long time, and the same applies to other pulses. But when we look at some growth promoters, and this may include Azospirillum, Pseudomonas, or the Bacillus consortia, and several delecensis. There are many bacteria, and due to hormone production processes or because they promote the use of a better use of nutrients or root growth, we can see they have an important impact on yield. And in order to overcome the barriers to the adoption of this technology, we just go for 1 or 2% of genetic gain in one hybrid. This is crystal clear, but so we are achieving results that are consistent of many years with many trials, field trials, when things are done as required by a biological. And actually we get to fertilization, nitrogen to promote growth. No, because it's another organisms, trichoderma, and this works on 2 areas. It promotes growth and it also controls diseases. But there is a lot of information so as to break that psychological barrier to adoption, to technology adoption, because the point is it should be properly used to achieve the results we see on the screen. And if we talk about a different challenge, which is much more interesting, the use of gram-positive consortia in leaf application or foliar application. We talk about biologicals, and usually we think about applying them to the seeds, but there is a whole world where I can use endospores to control diseases and achieve a very important impact on yield and productivity. And this is a statement which I believe is revolutionary nowadays. This gentleman, who was Ukrainian, 50% Ukrainian, 50% Russian, because he grew up in Kiev, and when it was part of the Russian Empire, he is the father of agricultural microbiology, and he taught us that the future of agriculture may be possible only if agronomy works hand in hand with microbiology. And this would be the end of my presentation. Thank thank you because both of you have completed your presentations in time. There is time for questions. At the very beginning, I didn't share my phone number. Nobody has submitted any questions, so if you want to ask any questions, there is a rolling mic. As the assistants are not here, I will give you my mic. I believe you probably define the formulations to the barriers to new formulations. Maybe they are good 1 year, not good the following year. Am I using them properly or not? Well, but there are still some marketing or commercial barriers. The distribution systems for agrochemicals are not paying attention to the sale of bioinputs, because we farmers realize the marketing or the sales pitches for bioinputs are not as good as those for traditional inputs. A chemical product is sold easily, but nobody sells biologicals really strongly. Let's look at the glass that is half full. I have an opinion and I have a statement too. Chemical companies are educating their sales forces and the technicians about the use of biologicals and about the proper management of biologicals. There is a change here. I can see this clearly. It's very difficult in their marketing equations. It's difficult to get the salespeople to talk about biologicals, but some companies are already developing specific teams to focus on these sales teams, and some others are taking part of the traditional salespeople to spread the message of bioinputs. My personal opinion now is that there is an issue in how we communicate the importance of biologicals. We, the ones who use them, and we believe we are all formatted except for the very young people, but we are very used to using chemical terms, the chemistry language. So as we understand triazoles, at the end of the day, the diseases are controlled by iterine, sulfactrine, which are produced by a living being. But we have an added value, because if that being is properly managed, we keep it alive in the plant structure, in the root, the leaf, or within the plant, and it keeps on producing that product. And therefore, the control efficiency remains in time. So we have companies that are setting up specific teams to sell biologicals, so they're training the regular teams. And on the other hand, we who are in the academia or in other companies, well, at the end of the day, we should spread the message that promotion or the control is carried out by a molecule. But this molecule was created by a living being, a bug or a bacteria, whatever, and not through chemical methods. Which are the barriers to the adoption of technologies? Barriers by farmers. Somebody asked about the distribution channel. Is there a difference in the barriers that different people face when we talk about technology? Yes, it's sort of difficult. Distribution, well, maybe an advantage we have is that as we work on microorganisms that produce spora, distribution is easier because we have solid and liquid products, and we talk about bacteria or UFC per milliliter, which is what is reported. This message, the products are arrive to the destination in good shape and they are stable in the long run. So our biologicals have many advantages. I would like to say something. I don't have much to say about your question because I am not experienced in the marketing, but one of our most important partners in our company is a company that manufactures chemical fertilizers. We have been working with a company that has a long-standing history in the field of chemical products. However, they opened up the doors for us with a kind of spin-off of Agrifert, and Agrifert has understood the outlooks, and they called us to come and partner and work with them because this is the future. It's a changing paradigm in the area of marketing, distributions. So I believe this is a transition time. I have another question by the floor. What do you think about the endophyte diazotrophic bacteria, Burkholderia and others? Could they stack up effects with PGPR? Do you have any trials in the central north of Argentina? Well, you could start for once. Come on. Well, I won't be here forever to help you. Well, this question, well, there is a bug, Methylobacterius symbiotics, that has attracted a lot of interest lately. These bacteria, besides being nitrogen fixation bacteria, they have other BGPR capabilities. In microbiology, we find this. These bacteria, Burkholderia, Paraburkholderia, Methylobacterium, well, what is important is not so much the name, but the role they play. So these bacteria have a role, and which helps to capture nitrogen from the environment and turn it into ammonia. I apply something on the leaves. I have to prevent stress from appearing. I have to get this bacteria into the plant, so it starts with nitrogen fixation. I have a lot of experience with Lupanesobacter. I've been working on this for many years. I worked with Mrs. Owenheimer. She was aware of the Haber-Bosch process. She knew about Haber and Bosch, but Glucocetabacter has been shown to be very good for biomass production and nitrogen capture. We didn't know how sugarcane was able to produce so much biomass as the soil didn't have so much organic matter or nitrogen. And there we found, Joanna found out how this happened. We believe there is a window of opportunity here. I don't think we are at a time to replace one by the other, because some people are saying, well, we are going to replace the units we have, the units of nitrogen we are using nowadays, but I believe we can change nitrogen by 15 or 20% and get important changes in biomass and yields, especially in grasses. Good afternoon. I'm Gustavo and Walter. I'd like to know, being a technical consultant and a business consultant, we have our own opinion, but regardless of the brands and the portfolio of biologicals in the market, I'd like you to tell us which do you think is the biological package which has the highest impact nowadays that could be used as an entry door to share this information with the farmers who are reluctant to use this new technology, that is starting with a technology that will achieve a good impact and a secure one. so that the farmers will, after that, be open to learn about new products or carry out different trials? Well, in our experience, for example, using microbial consortium with the current technologies, we achieved very good results. As I was telling you earlier on, the consortium, the B4 we developed when it's used together with another product and expands the availability and improves the microbiome for the plant, this increases yield quite a bit. So if you share that information, it should be easy to convey the message to the farmers who are reluctant. We believe that consortia have many advantages. When I talked about microbiome earlier on, we have found the particles produced by the roots. The root exudates have signals that attract. Each plant has a specific microbiome. The microbiome of a soybean plant is different from the microbiome of a wheat plant. So the evolution by the biologicals is related to microbial consortia. So using technologicals with the current chemicals should be very positive. Just to complement what Walter has said, I was in your shoes. So when you are in the first line, you are in the first line. And you're not the general who tells people go this way or that way. No. But just complementing what he said, I believe a good entry point would be to talk about the function, the role. Talking about the function, the role. If you say, I have to fertilize the crops, I have to provide nutrition, it's clear that that plants need nitrogen. Look what happens when I use in a grass methylobacterium that provides nitrogen, or in a pulse, a rhizobium that provides nitrogen. When you show these figures or this information, sorry for talking in lay terms, so to say, about this. Look, here you have a solution which is environmentally friendly, but besides, it's also profitable. And what are you looking for? Well, do you want to use urea or do you want to have nitrogen? So if you want to have nitrogen, this is a tool that enables you to use that nitrogen in a different way with all these advantages. And when you talk about pest disease control, most of the success of trichoderma, when you look at the past, well, it has grown quite a bit, is that in our way of thinking, now I'm wearing the farmer's hat, I know that wheat has to be treated with different products or whatever, carbamazepine or whatever, you name it, but I use treatments. There are many ways of treating wheat. Look, in this whole portfolio, you have this tool, which is called trichoderma, and not difenoconazole or whatever. So, and if you use it in this way, you get the same results, but it has a bonus track because the bacteria, just like all living organisms, don't do just one thing, they also do more things. So maybe in this season in soybean we will need to use seed treatments, but sometimes it's not necessary. So these bacteria promote growth. So we tell them, you take this, but you use this, but you have this as a bonus track provided by the microorganism. I don't know if this was your question, but well, I just wanted to say this. Thank you. Can you hear me? This is a question submitted by WhatsApp, and I will merge it with a question I thought of as we met several times to prepare this session. Our inputs, innovations, or disruptive technologies. My question is, could we do with bioinputs what we have been doing with chemical inputs? Could we overuse them? Is there a risk of overusing them? Will we stay until 10 p.m.? No, I have to leave earlier because the parking closes at 9:30. I believe, this is my opinion, because the question is asking for my opinion. I don't have the data so as to share them with you and show a clear trend. But I believe, number one, plants have evolved together with the microorganism. So when we think about microbiology tools, it's hard to understand why we have plants on one side and the microbiome on the other hand. Well, because this is the way we have been thinking of in the past, thinking of bacteria. So I don't grasp this fully. So they evolved together. So we cannot understand plants without the microorganisms, and we human beings could not be seen without our metagenomes. Secondly, I don't believe microorganisms will be able to get the results we were not looking for with chemicals, because in the soil we have many microorganisms, and since we're just introducing 2 million bacteria, 100 million bacteria in 1 hectare. When you multiply times 20 centimeters, you get a large figure of microorganisms which are there, which have been there for a long time. And they are staying there, and they're fighting for water, nutrients, air. So the soil reaches a balance with microbial flora, microbial population, and the soil stops the unwanted growth, because when there is a plant or where there is a root, it's like everything looks like Paris. All the issues are solved, but the plant, we want it to die, or because they die on a yearly basis because we do the harvest, and the carbon flow disappears. So all of us, those who were in Paris, are now being bombed in Kiev by the Russians. So, well, I don't think that will happen. I believe there's a microbiological revolution ahead. That is, we should use cells to produce molecules. When we look backwards, and we see many molecules used nowadays based on synthesis, these molecules were biological. Earlier, fluoxanil was a metabolite of a pseudomonas. Spinosad was a metabolite of a different microorganism, and so on and so forth. So the challenge looking ahead is to use the cells to produce biomolecules to achieve the same goals with a lower environmental impact than the one we get from the use of chemicals. Well, I believe there are no further questions. Besides, Gustavo has to get his car from the car park. Thank you for your excellent presentations, and let's